Beautiful bling bling microphone, so that should work. Excellent. So welcome everyone. So my name is Johan Sandin. I'm Chief Scientific Officer and also one of the founders of AlzeCure Pharma. And I'm going to present a bit about our company, which is also based here at Novum, for you today. So our company is working on Alzheimer's disease and pain and developing novel therapeutics for these areas, which are areas of huge unmet medical need. All of us founders came from AstraZeneca at one point. So the closure of the CNS site led actually to the start of this company. The company itself was founded in 2016, but the organization stems back to 2012 when it's actually founded as a foundation. We have three project platforms that are all small molecule based. One is called Alzstatin, which is a preventive and disease-modifying treatment for Alzheimer's disease. We have then NeuroRestore, which is a symptomatic therapeutic with the potential for disease-modifying effects. And then we also have Painless, which is a platform containing two pain programs, one for osteoarthritic and the other one for neuropathic pain. And I'll go through these a bit more in detail later. As a company, we are listed on NASDAQ since November 2018. We are a research and development company with the aim to develop our programs up until phase 2 clinical trials and then out-license the programs to gain incomes. So a bit about our history as such. The company, as I said, was founded in 2016, but the foundation, which is really stems back to 2012, when 5 of us co-founders started this together with the Swedish Alzheimer Fonden, as well as Karolinska Institutet and Bengt Winblad here, to initiate the AlzeCure Foundation. So we had some, what we thought was really great ideas, and we got the support from various funding sources such as Alzheimer Fonden, Alzheimer Drug Discovery Foundation, Swedish Brainpower and others to initiate the work and execute our ideas. And it eventually led to a full discovery and development work with the NeuroRestore program, which I'll talk a bit about more later, which is currently moving into phase 2 clinical trials. So that's gone from idea all the way to clinical studies, which has really been a fascinating journey to where we are today. So this is our pipeline. As you can see here, you have the 3 different project platforms to the left. For the NeuroRestore program, we have ACD856 as our lead molecule, which is now moving into phase 2 clinical trials. This is a positive allosteric modulator of TRAC receptors. And I'll talk a bit about that later on. Alzstatin, which is a gamma secretase modulator. That's currently in preclinical phase, moving through the safety and toxicology studies before moving into man. In the pain program, we have ACD440, which contains TRPV1, ACD440, which is TRPV1 antagonist, and that's currently moved through phase 2A clinical trials with positive data. And then we have ACD137, which is a negative allosteric modulator of TRAC-A receptors, and that's also in preclinical stage, going through the safety and toxicology findings before moving into man. So I'll just start with our 2 Alzheimer's platforms, the Alzstatin and the NeuroRestore, which is really aiming towards 2 different stages of the disease. As you know, Alzheimer's disease is a disease with a long pathological process over decades, actually. And Alzstatin targets really the early stages of the disease, while NeuroRestore targets the more symptomatic later stages of the disease. Now, Alzstatin, the first program, that targets the amyloid cascade, which you've probably heard a lot about, certainly with the advent of these new antibodies, the amyloid antibodies, which have reached the market. Now, as you know, these characteristic amyloid plaques, which are found in the brains of Alzheimer's patients, are formed by a small peptide called A-beta, which is cleaved out of a larger protein, and then starts aggregating into larger and larger aggregates, oligomers, protofibrils, fibrils, and last, these amyloid plaques. And what alstatin does is actually reduce the production of these small building blocks called A-beta 42, and thereby prevent the aggregation of these larger aggregates and eventually forming the plaques. So it's really targeting the initiation site rather than the antibodies, which actually scoop up these different fragments, really depending on what the antibodies will bind to, either fibrils, oligomers, or plaques. So what we've seen with the compounds that we have moved through the platform is that we have a very significant and potent effect on the formation of these toxic peptides, the A-beta 42. So as you can see here, we can significantly dose-dependently reduce the amount of A-beta 42 in the brains of mice with more than 60%. And interestingly, with this type of strategy is that what we do with the molecules is that we decrease the amount of these aggregation-prone and toxic A-beta 42 and 40 species, while at the same time we increase the amount of these shorter peptides, which have actually shown to have beneficial effects preclinically as well as in clinical studies, actually. So it has a dual effect, you could say. And this program has also been kind of validated by Roche, which are currently in phase 2 clinical trials now. Then we have the NeuroRestore program, which is a positive allosteric modulator of TRK receptors. And here we're targeting neurotrophin signaling. So neurotrophins, the most well-known, probably NGF, nerve growth factor, and BDNF, brain-derived neurotrophic factor. And they do bind to the receptors, the TRK-A or TRK-B receptors, and mediate important functions, downstream functions in the brain, such as neuronal proliferation, neuroprotection, synaptic plasticity, effects on mitochondrial function, but also certainly plays a really important role in cognitive function. And these systems become dysregulated in disorders such as Alzheimer's disease and Parkinson's disease. And what we're trying to do with our compound here is trying to normalize the function in this system and thereby enhancing synaptic plasticity, improving cognitive function, which we've also been able to show preclinically. Given its effects on neuroprotection and mitochondrial function, anti-inflammatory function, there's also a strong potential for disease modification here, which is another benefit of the system. So there are actually, as I mentioned, both cognitive beneficial effects, but also disease-modifying effects with this strategy. And we have tested these compounds also preclinically, and we've been able to show that the compounds affect both learning processes, storage processes, as well as recall of previously stored information. So it seems to affect all of the different cognitive processes leading to memory. And this is just an example here showing a study we did in aged animals. The animals are at an age which would represent about a 75 to an 80-year-old in man. And here we've looked at their memory, either in a fear conditioning task, either 1 day after learning or 11 days after learning. And we compared it to that of young mice. And a high bar here means a good memory. So if you look at the young mice, they perform equally well at day 1 as they do on day 11. The aged mice, here you can see a tendency to decline already at day 1, and at day 11 they are performing at chance. However, if you've given the aged mice the ACD856 compound, you can see that they are as proficient as their young counterparts. So it can certainly revert these age-induced cognitive impairment, and we've done this also in other models So a clear cognitive benefit with these compounds. We have then further moved on to clinical studies with this compound, shown that the compound is safe and well tolerated. It has an excellent pharmacokinetic profile, so it's suitable for once daily dosing, which is excellent for this group of patients. It also passes the blood-brain barrier very efficiently, and we've seen that it engages neuronal circuitry in the brain relevant for both cognition and depression. So we're now preparing for phase 2 with this compound. And actually, just earlier this year, we received a European Innovation Council grant for this phase 2 clinical trial with this compound. So that was really good news. Next, moving into our painless program. So here we have 2 programs targeting really 2 different areas: osteoarthritic pain, again, a very large patient population with a TrkA NAM program, and neuropathic pain with the ACD440 program. If we start with the TrkA NAM program, so this is a negative allosteric modulator of TrkA or NGF signaling, both of which have been shown to play a really important role in pain signaling. And it has a strong validation also with the fact that anti-NGF antibodies have been into late-stage clinical trials and shown a really step change in efficacy when it comes to analgesia. But they've also shown in a smaller proportion of the patients side effects that are rapidly progressing osteoarthritis. Now, we believe that the reason for these side effects is that these anti-NGF antibodies will target 2 of the downstream signaling pathways of NGF, both the P75 signaling and TrkA. Given that we have a TrkA selective mechanism of action here, we aim to maintain the clinical analgesic efficacy but avoid the side effects. This has really been also a holy grail within the pain area to find compounds that are so-called TrkNAMs. I think I would say we have strong interest from pharma industry already now, even though we're at an early stage. And we have also conducted here studies in preclinical models. This is showing the data from the rat MIA model, where we did see, which is a really validated model for osteoarthritis, I would say. And here we could see a strong analgesic and potent analgesic effect with ACD137, which is our lead compound, similar to that of an anti-NGF antibody. At the same time, we did see a protective effect with these compounds on the cartilage damage that is induced in the MIA model. So very potent and good effects with the compounds. And here we are, as I said, kind of moving through the preclinical safety and tox testing before moving into man. ACD440, which is the last program, this is a program that emanates from Big Pharma. So this compound was originally developed as an oral compound, targets the TRPV1, and the TRPV1 is involved in heat sensation, so the sensation of heat, but also temperature regulation. So when this compound was used as an oral or systemic treatment, what they did see in the patients was an increase in body core temperature, obviously not pleasant, but also they got an insensitivity towards heat. So if they drank hot coffee, they would burn themselves because they couldn't feel the heat itself. So when we in-licensed this compound from AstraZeneca, we reformulated it into a topical treatment, so a gel that you can put on the skin, because and has since been driving this for a different indication, peripheral neuropathic pain. So, for example, post-herpetic neuralgia. So here, the painful area is in the skin, and what you see in these indications is an upregulation of TRPV1 receptors in the skin. So our idea was to maximize the concentration of the compound by giving it locally, but avoid systemic activation and avoid these side effects. And that's also what we've seen now, both in the phase 1b trials in healthy volunteers, but also in the phase 2a trials in patients with neuropathic pain. So here in the phase 2a trial, we did a study in patients with chronic peripheral neuropathic pain. And we had, what we did see was a pain reduction of approximately 50% on evoked pain. So a very good effect. And we also were able to test our bedside test to identify patients which would be eligible for this type of treatment, which also worked very well. And it was also well tolerated and no side effects. So we're now preparing for the next phase 2 here. So I would say we have a very broad pipeline, which is really targeting areas of great unmet medical need. And I would say we have a very strong team, and we are now, I believe, 11 employees full-time. We have about 30 consultants working with us. And we also have platforms with first-in-class properties. And we have invested in several candidates in parallel, which I haven't shown you, but within these 4 programs. So, and I think we're really proud that we've evolved from a discovery company with really from ideas to where we are right now with phase 2 programs in clinics. So great fun and an excellent journey to do actually. So with that, I'll take any questions and thank you for your attention.